Dr. Mumberg introduced himself already during his talk. I would like to shortly introduce the other panelist. It's Dr. Strepart from Frankfurt. He is a cancer biologist. And Dr. Lang from Essen. He is a head and neck surgeon with a strong interest in immunology. And Dr. Singh comes from the Memorial Sloan Kettering Cancer Center. He's also a surgeon and has a very strong background on molecular biology. My name is Henning Bier and I'm coming from Munich. So, I would perhaps like to start with your last slide, because in head and neck cancer, we very often face the problem that when we do clinical trials, that we have very heterogeneous groups which are compared. And now, seeing your last slide, your idea is to even look for more divergent stratification of patients. So my problem in thought is just that we will have no conclusive answers because we don't have the sufficient patients cohorts to make a good point on what we are doing actually in this kind of treatment.
Yeah, thank you very much. If I may reply to that. That's actually the reason, but of course certainly escaped the attention. That's why I'm careful of using the term stratification biomarker from the beginning and rather talk about predictive markers. Because if I'm allowed to, as a non-clinical scientist, kind of to suggest for clinical trial design, the point to me would be not necessarily, at least not in early trials, start immediately to stratify, but just include into the trial design the analysis of such markers in order to learn about. So the point is, if there is now any one of these new agents, you refer to that, there are many agents coming to the clinic and all look very interesting or exciting, but then you have to figure out how to apply them. The point to me is, from the very beginning, take the extra effort and kind of take samples, take a biopsy of the tumor or circulating tumor cells, whatever, and we can discuss about that, do extra imaging in order to understand or have a chance to understand the mechanism underlying either a response or non-response. And this is very crucial. It is very clear that when you say, ideally I would take, let's say, either a biopsy or sampling before the actual treatment and then maybe under treatment or so. This is a heavy burden on the patient and of course an extra effort then also on the clinician, on the oncologist. But I think this is really crucial in order to make progress in that field. Because often it happens like we had with the Iressa in the Tarceva field. Iressa was approved without having any idea of that. Now then when it turned out in the phase 3 studies, at least for Iressa, that the drug was not effective, then retrospectively people would go back and analyze it in more detail and find that out. Would have it been known beforehand, it could have been used much more to the success of patients.
So your suggestion would be that we take care of a very good tumor bank, tumor collecting, sample collecting, providing all measures which are sufficient to retain DNA, RNA, serum, PBLs, and so on. So this would be a direction you would suggest?
Certainly, I would suggest that. And the point is, often, and this is now from the perspective of a pharmaceutical company, kind of then trying to find the optimal use for drugs, it is really in the best interest. And often the point is, it is not only sufficient, let's say, at the point of primary diagnosis. So in breast cancer, you had a diagnosis maybe 8 years ago, and then the primary tumor was removed. Now the patient comes back. Now this tumor now may look significantly different with regard to its mutations, but this is crucial in order to understand the response or non-response. So therefore, kind of in conjunction, of course, in close coordination and collaboration, then with academic research, pharmaceutical industry, really let's try to make the most out of it. And I would say, yes, in major centers, there should be no patient be treated, at least not in clinical studies, without sampling and archiving of those probes in order to be able to look for that.
Right. Dr. Strepart, considering the redundant organization of the malignant phenotype, do we have to realize that oncogenes perhaps are not the best target we have? What about other targets like suppressor genes or microRNA genes, which are getting more and more important? Do you see there any kind of escape pathway from all these problems which we have heard of in the lecture of Dr. Mumberg?
Well, I think in general, I'm coming from the field of protein kinases, which are a major section of drug targets. There are, in addition, several other targets coming up. For example, heat shock proteins are targets. There are pleiotropic effectors. HDACs are being considered. So there are a lot of different types which are depending on the tumor type. So let's say Gleevec was from my point of view, a clinical triumph, since BCR-ABL is the major driver of this oncogenic transformation. But in most types of cancer, we have not a single driver. We have, let's say, in solid tumors, a selection of 6 to 8 different oncogenic alterations. And I think in terms of treatment and in terms of diagnosis, we have to take care of all these. We have to consider more than one oncogenic transformation. We have to consider different pathways. And either with multi-targeted inhibitors or with appropriate combinations, I think we have to target different ways of malignant signal transduction.
So, for example, do you see synergistic activity if you combine several anti-protein kinase agents? In preclinical models, is it additive effect? Is it synergistic effect?
Well, there are different or a broad spectrum of studies showing that there are synergistic actions between agents targeting the cell cycle or agents targeting especially angiogenesis or receptor-related targeting or receptor-related signal transductions. There are many examples in the literature.
So taking these 2 points together, that would be trying to identify or profile the patient's tumor and then mixing a good cocktail for this particular cancer. Who will pay for this?
I think I would ask it the other way around. Can we afford not to pay for it? The point is, if we look at this point in time about the incidence and the mortality from cancer, I think it is obvious that whereas in most other disease areas we have made significant and often traumatic progress, just think about infectious diseases or so, in cancer the progress, although clearly there, has been modest or limited. And since the population is, at least in the Western Hemisphere, clearly getting older, thereby increasing the risk of age-related cancers, which there are many, I think this is an issue which cannot be neglected. And the question is, to me, not whether we can afford it, but we have to find ways how to make it affordable. And therefore, we need maybe new strategies and new approaches, and maybe we can discuss that later, how to accomplish that. But I think I fully agree, of course, the costs are exaggerating, but this is not due to the fact that people want to make it expensive or somebody wants to make a lot of money. But the point is simply right now, just doing trial and error is unaffordable. Make it very clear. In the development of a drug, the by far most cost-expensive part are the clinical trials. And why is that? Because we have no idea or often limited knowledge when we go into those trials, and therefore we need then kind of, 2,000 patients in a clinical trial with a placebo group together, because we just try to find the needle in the haystack. If we would be able to define better the patients who are likely to respond, we could reduce the costs. Maybe, hopefully.
Hopefully. Yes, please.
Well, I can just agree to this statement. I think the attrition rates just in oncology are about 95%. So just 5% of the drugs are really approved by the FDA or here in Europe by appropriate organizations. So I think before these drugs come to the clinic, we need appropriate animal models. We need different animal models to get a better prediction of side effects. Right. I think the development of appropriate biomarkers is also a very important field to reduce or to improve the efficacy in the clinic.
Right. Dr. Singh, we have learned from molecular biology that oncogenes normally, if they're activated, should drive cells into apoptosis. So are there strategies to try to combine these two fields, that on the one hand we cut off the stimulation, and on the other hand we will help the tumor cells to enter into programmed cell death?
I think, sorry, I'm losing my voice, so hopefully you'll bear with me.
What did you do yesterday evening?
No comment. So I think I just would like to step back one step. If you look at the way most solid tumors develop, there is a panoply of genetic events that accumulate. And if you look at modeling systems, there's probably about 10,000 different genetic events that accumulate in a single cell, many of which are not in genes. They may be in promoters or even non-coding regions. And the problem is not identifying these targets, because we've identified EGFR, we've identified VEGF, we've identified all these other things that potentially work as oncogenes in these tumors. The problem is identifying what are the critical oncogenes that will drive this dependence. And if you ask someone 5 years ago if there is a critical oncogene in lung adenocarcinoma, specifically the bronchoalveolar type, the answer would be no. But now we know through experimental confirmation that EGFR is indeed a critical oncogene in these tumors. And there is a resistance that develops over time, but while you're treating, the results are dramatic. I think it's very important to point out that you start the drug on a Monday and you can have a lung full of tumors, and by Friday you can repeat the chest x-ray and 90% of the tumors are gone and the patients are completely functional. And this was one of the initial papers that came out was from our group at Memorial, and this was the patient in which the EGFR mutation was identified. And we were fortunate to have banked the tumor to be able to identify it. But really what drove the identification was not the fact that there was response or lack of response, was that there was a development of a new analytic tool or availability of a new analytic tool to allow for detection of mutation. And it wasn't a targeted analysis that was done. It was a kinome-wide sequencing effort that was done, and EGFR turned out that was the only one that we can detect in that single patient. So the reality is, I think you're absolutely right. I think we need to bank these tumors. We need to allow technology to catch up, and we need to identify the specific targets and the specific oncogenes that drive the progression of these tumors. And the other reality is, as we can also take back from lung cancer, that each individual cancer will have not one agent that drives it. And so we need to understand that we're not going to have a magic bullet that's going to treat head and neck cancer. We may have 50 magic bullets, and we have to individualize the selection in order to achieve appropriate outcomes. That's not going to happen easily. I mean, that's not going to happen with available measures. Gene arrays, DNA arrays, sequence analysis are not going to be sufficient, and technology has to catch up before we can do that.
Okay. So your lecture focused on oncogenes. We also have tumor suppressor genes. I think the oldest targeted therapy is immunotherapy, and there have been great success now with prophylactic vaccination. You are looking for specific, more or less specific targets, antigens in head and neck cancer. So what do you think? Will we really get a sufficiently specific target for immunotherapy? And the second question, which is linked to this, of course, do we have the chance that, do we have a considerable chance that in a therapeutic setting, immunotherapy will work?
That's a very good question. The title of this session is Molecular Biology as Basis for Treatment. And as you point out, it's very important that we also apply molecular biology for better diagnostics. We have to identify more specific tumor antigens, target structures for a more specific diagnostic procedure in order not only to just maybe discriminate between scar